From Microbe TV, this is a special episode of Immune, an immune booster recorded on October 3rd, 2025. I'm Cindy Leifer, and you're listening to the podcast about the body's defenders against disease. I'm here in my office at Cornell University for another office interview where we learn more about the scientists behind the science. If you like these office interviews, Immune, or any of the other Microbe TV shows, please consider contributing. You can go to microbe.tv/contribute. So today, it's my great pleasure to be joined by Kristian Andersen, who is professor in translational medicine.
That's correct. Good to
at Scripps Research Institute. And I'm super excited about this discussion today because Kristian's focus on research is really on the complex interactions between the host and the microbe, and that's my favorite topic. And he's a global expert in viral evolution, but also a bona fide immunologist. We're going to talk about that.
T-cell immunologist, yeah.
T-cell immunologist. He did a PhD in immunology, and I think it's a great perspective on how immune responses and other factors like urbanization and other changes shape virus evolution and how that contributes to the development of pandemics. He's done absolutely groundbreaking work on virus evolution in multiple different viruses, Zika, Ebola, Lassa fever, and of course, COVID-19. I'm sure there's more. He investigated the source of COVID-19. It's brought up some interesting controversy. If you want to talk about that, we don't have to. And it also led to talking to a subcommittee in Congress. Not very many scientists can say they've done that. So that's very interesting. He's also been on TWIV before. So it's a really great pleasure to have you on Immune now.
Yeah, thanks for having me. It's great to be here at Cornell.
Great. So I was particularly interested because, one, you're here giving a talk today, but also relatively recently, earlier this year, you were one of the co-authors on a perspective article that I found absolutely fascinating. The title of it is Immunological Drivers of Zoonotic Virus Emergence, Evolution, and Endemicity.
Right.
That's a hard word to say.
Yeah.
Anyway, so I have lots of questions about that and how viruses and the immune system interact and everything. But first, can you just tell us your story? Like, what's your story?
Where do we start? Yeah, no, I mean, my story into science is that, as you mentioned, my PhD was in immunology. And I'll say, like, I'm not a classical scientist in the sense that I didn't grow up thinking I would become a scientist. In fact, I never really thought much about it. My family is not from a family of scientists. I'm the first one to go to university, for example, in my family. But I always liked challenges and questions and problems and, just trying to solve things, whatever that had to do with. And of course, science has a lot of that. I was working at the post office back in Denmark, for example. I'd been taking my high school degree, of course, a diploma, and also went to business school and went to the military for a while and, was in the Danish Royal Horse Guard, for example, doing that. And then I was working at the post office afterwards, and I was really just looking at, what do you want to do for the rest of my life? And maybe I was, work at the post office. It was a pretty good job. I actually missed that quite a bit. But then, super privileged, and in Denmark, anybody can basically go to university, right? Not only do we not pay to go and get our education, we also get a stipend. So I had the ability to saying like, okay, I always liked, physics, chemistry. So I wanted to study that at university. And then I went to University of Aarhus in Denmark and got my bachelor's degree there in molecular biology and biotechnology. As part of that, I did a year abroad in the UK at the University of Kent in Canterbury, where I met my future wife. And studying sort of biomedical science there as well. And that took me from there, and I realized this is actually pretty interesting. I like science. It's cool. Can we maybe make a career out of this? And started doing crystallography for one of my first projects, studying structures, EFTU, ribosomes.
Oh, so ribosomes, so more cell biology based crystal structure stuff.
Straight up, expressing proteins and E. coli, purifying them. This was from Thermophilus, so it's a heat stable protein. Amazing. You just basically boil it out. Do a couple of purification steps, and then you have almost clean protein, and then it crystallized pretty quickly too. And we complexed this EFTU translation factor from bacteria with an antibiotic, and we actually solved the structure of that within just a half a year or so. And that was my first experience to this. And I remember my PI, he was pretty excited about it. I didn't understand what was going on because I was like, yeah, sure. I mean, isn't that what's supposed to happen?
Crystallize proteins in 6 months? Sure, no problem.
It's like, and I honestly I just had no idea. Of course, science then turned out it's actually harder than that.
It's a little harder than that most of the time.
But I then went from the University of Aarhus and I wanted to study immunology because I liked the complex nature of the immune system, and there are many different questions in immunology. And that's where I applied for the University of Cambridge at the Laboratory of Molecular Biology, got accepted into their program, and then I was studying there for 4 years, focusing on tolerance mechanisms and very specifically regulatory T cells in mouse systems primarily. Part of this was understanding regulatory T cells during pregnancy, for example. They're required for that because you have the paternal antigens are being expressed. That obviously needs to be suppressed, otherwise, it will get rejection of the fetus, and obviously that does not happen. And regulatory T cells play a pretty key role in that. And that sort of got me interested in the evolutionary aspects of that because, you have the evolution of mammals and marsupials, from vertebrates, right? And the question is, how does that work in terms of the evolution of regulatory T cells themselves? And they have a transcription factor, lineage-specific transcription factor, master switch called FOXP3. So I looked into the evolution of FOXP3 and how does that evolve over time, and could show how going from birds, for example, that obviously do not have, placentation. So the evolution of FOXP3 along that line, and then ending up with what we see today as being FOXP3 and regulatory T cells in mammals specifically.
So birds have regulatory T cells
no,
too?
So they probably don't. Well, so at the time where I did the investigation of this, and we have a paper on this where we described that actually we did not find FOXP3 at all in birds. FOXP1 and 2 and 4 were there. But FOXP3 was not there. I will say, though, that, and we also don't see it in fish, for example.
Okay.
But we see, orthologs that look a lot like it. And I will say, like, the sort of incompleteness of the time of the genome, so it was the chicken genome in particular, is that they probably do have FOXP3. Whether they have regulatory T cells, maybe, and probably there's been some papers published on this. It's just maybe. I haven't studied regulatory T cells for, 15, 20 years now. So I don't know, but that got me interested in the sort of more evolutionary aspects of things, right? And that's where, so when I was looking at my postdoc, where I was like, look, I don't actually want to continue to do immunology. I don't want to do experiments in mice.
Why not?
Immunology.
Why do you want to give up immunology?
I needed to try something else. Okay. At the time, Illumina sequencing, for example, or Solexa sequencing, as it was called at the time. Was starting to become more of a thing. I like data science, I like computers, and I was like, look, I want to do something which is a little bit more mathy, a little more data science, and then maybe some of the genomics and things like that over it. And I meant that for my postdoc, I just, I was looking for labs that were, doing mathy data science type of stuff, but with evolutionary questions on top. And interviewed with a few different labs, and of course ended up in Paris Sabetti's lab at Harvard at the Broad Institute. And Paris, who's amazing scientist, right? And Paris had been doing these selection scans in humans and found all these interesting genes that appeared to over evolutionary history in humans had been under selection, and probably with viruses being the selection force here. So one of the genes that she had found was a gene called LARCH, which is a large gene, but it also happens to be part of the entry pathway of Lassa virus, and she had found it to be under selection in the West African populations. So my first part of my project was to look into that further on the human side, human genomics, look at selection scans in humans.
When you say selection, you just mean like certain populations
it's just certain alleles.
Have different alleles in other populations that they're more likely, for example, if it's a loss of virus receptor, they're more likely to be infected.
Correct. Yeah. Okay. So basically the idea here is, so when we're looking at human evolution, right, is that you have different alleles, variants of genes, right? Where, for example, you can imagine in large, which might, be a restriction factor in the way in which Lassa can or cannot enter cells, can or cannot enter specific cells or whatever that is. Lassa is the receptor for Lassa, the main receptor for Lassa is alpha-dystroglycan, and large is sort of downstream of that. And what Paris again had seen was that basically different alleles of large that look to be risen in frequency in the West African population specifically. These are called long haplotypes. Basically, the gene has a long haplotype, which means that over time, the haplotypes will break down, right? Just because that's what happens during meiosis, whichever. But when genes are selected, is that they're being driven up in frequency quicker, and then take these long haplotypes with them. So they look like they're old in the sense that they're high frequency, but they look young because they have these long haplotypes. And she had found these variants in the West African population. And then she had noticed the connection with, again, Lassa virus. It's part of the entry pathway. But really importantly, Lassa virus is endemic in the West African population. So she was interested in understanding, could there be a selection pressure, and could large and these variants of large be a restriction factor, as we're talking about Lassa virus in particular. And my interest then turned to the virus, where it was like, okay, so what do we know about the virus? And at the time, not very much from a genomic perspective. So our first foray into this was, just investigating the evolutionary history of Lassa, understanding its spread across West Africa, its age, it's a very old virus. And we did that by sequencing Lassa from patients in West Africa, from rodents that serve as the reservoir. Mastinus natalensis is the multi-mammoth rat, is the reservoir for Lassa. So we could, get it from those and sequence from those animals too. And that allowed us to sort of, show the evolutionary history, the origin, if you like, of Lassa virus and its spread across West Africa. And of course, with the Ebola outbreak happening in 2014, that then became a focus because we were working in West Africa, in Sierra Leone, Nigeria, but Sierra Leone at the time, of course, got hit hard by this. And we were there setting up diagnostics and helping them with, my colleagues there, myself and Stephen Geyer, another scientist in Paris's lab, traveled to West Africa and set up diagnostics and made sure that they could diagnose patients if they got any. And then, of course, on top of that, we coupled the sequencing too, so we can understand the West African Ebola outbreak. For example, did it spill over once? Did it spill over multiple times? And then how did it evolve as part of that outbreak itself?
And spillover. So we're talking now about there's a reservoir that has the virus, and then somehow that's coming now, being able to replicate in another individual or population like humans. And when you say spill over once or spill over twice, how do you use the information that you get from all these virus sequences to try and figure that out?
Yeah, so, viral phylogenetics and phylodynamics, where we have, viral genomes sampled from infected individuals over time. We have a location and we have a time associated with a virus genome. And because the virus is mutating over time, it sort of creates this family tree. And on top of that, we can fit a molecular clock, right? Because viruses have sort of a clock-like accumulation of mutations, similar to we age. And unless pandemic happens, the aging is relatively constant. And pandemic happens, and it's accelerated a lot. Some of us, but in the virus, it's quite stable, right? And we can then use that and say, okay, now we have the number of mutations that we see over time. We can fit a clock on that. We can go back in time, and we can understand when it started. So for Ebola, for example, we knew the first reports dated back to December of 2013, for example, although we only detected it in March of 2014, right? So about 3 months later. And we could show based on the genomics that that's exactly when it started. Like we, again, fitting the molecular clock and doing that on late December was the start of the outbreak itself. But we can also show that it basically goes back to one common ancestor. And not two, like we see with SARS-CoV-2, for example. We have two early lineages, lineage A and lineage B, which are highly consistent with this virus spilling over twice. For Ebola, it was clearly just once in Guinea and linked back to probably the first cases that were then ascertained later on. But one of the important questions we were interested in is that the messaging around Ebola virus during that time. This was the first time Ebola virus was found in West Africa. It never caused an outbreak there before. Always been in Central Africa, Middle Africa, right? The DRC, for example. And now they had a massive epidemic of Ebola, much larger than what we had seen previously. And the messaging around this, for example, was that, markets were closed down where they were selling, things like dried bats and other things that they use for consumption, right? With the fear that people were getting Ebola from bats. But of course, that's not how people were getting Ebola during the epidemic, right? They were getting it from other people with Ebola. The first patient probably got it from a live bat and got into contact with feces or whatever that was from the bat and got infected with Ebola as part of that. But then once the epidemic goes off, then it just goes off from there, right? So it was really important that our study could show that actually it's just a single introduction that went into the human population. It started back in December 2013. We're now in May of 2014, June 2014 or whatever. People are not getting Ebola from these markets in which, bats are sold, for example. And that's not the risk, right? And in these communities, taking away like food sources like that is not without consequence, right? I mean, the trading is important. It's the main protein source for a lot of people too, right? So being able to use the science to show that actually that's not how people are getting Ebola, right? It's just that other people have Ebola and they get into contact with that. So we need to focus on things like washing your hands and like having good hygiene around, Ebola patients in general. And I think that was important because, the Ebola outbreak ultimately came under control because of the local community sort of really taking it seriously and understanding the kind of things that they had to do in order to prevent onward transmission from there, right? That took a long time and it was a devastating outbreak, right? It's still the biggest epidemic that we have seen of Ebola. But yeah, a lot of that work was done there. And then that obviously took us into Zika later on and then SARS-CoV-2 and other viruses when I started my own group.
So this idea of controlling the bat consumption and that potentially then causing nutritional deficiencies, and those are things that also change susceptibility to viruses. And so can you talk now a little bit, coming into the perspective, like how do those different factors, so environmental, nutritional, immune, put various different pressures on the virus, and how does that change how viruses evolve, how they can get into populations and so
and I think this is, because I've gotten very interested in like the viral evolution side of things, right? And the sort of really interesting part about that is that, it differs depending on which virus you're talking about. Which obviously that's not a surprise, right? But, it really differs in quite dramatic ways. So if you're talking about, something like SARS-CoV-2, for example, is that, of course, in the early stages of the pandemic, this virus all of a sudden found itself in humans, right? It's like, what is this host? I haven't seen this one before. It's like, but there's a lot of people. There's no immunity here. Wow, I can just keep spreading. But, then you have this sort of selection pressure being like, but it's not great at spreading among humans, right? But it's certainly good enough to get off and cause a pandemic, right? But what we saw during that first year is really that, the virus evolved a lot where they're just inherent transmissibility of the virus, which is captured in this R0, right, the basic reproduction number, just increased, right? It just became a quote unquote better human virus, right? And that's what we first see in sort of the first crop of mutations like D614G and other mutations that were talked about at the time, right?
And they change the ability to bind to the receptor.
Yeah, they sort of tweak the ability of the receptor, it tweaks the ability of the polymerase to work efficiently in humans, the stability, the sort of compactness of the spike itself probably also changed as part of this, right? And it just has to do with how you have human ACE2 receptor, and it's pretty good already, right? But it can still get better. You had the furin cleavage site, for example, of which a lot have been said is that there was a lot of evolution around that too, right? Because it was, when it first came into humans, it was pretty suboptimal, right? It's not a great furin cleavage site. And there was a lot of evolutionary paths in which the virus could optimize on that. And then those viruses would just have a higher fitness and would ultimately take over in the population. And we saw that in the very first stages, the first year or so of the pandemic, right? But then, of course, that changes by the time that, in some cases you had, in some areas of the world, like Brazil and South Africa, where you had a lot of really significant and big waves, first waves, in which a lot of people were infected. In fact, in some of these areas, the local areas, probably most of the population, like Manaus in Brazil, and like the Western Cape, for example, in South Africa, right? Where probably most of the local population got exposed and got infected with SARS-CoV-2 in the first wave. And then you see the emergence of variants, right? And you see on one hand, you see like Alpha, for example, which is first detected in the UK. And Alpha just looked like something that was even better at transmitting among humans, right? And ultimately Alpha took over the whole world.
Yeah.
But then you saw things like Gamma, for example, that was in Brazil, right? Again, associated with these communities in which they had a lot of population immunity, and now it's causing a new wave. And Beta in South Africa, where again, Beta was first detected and now causing second waves in the population in which they had a lot of immunity from infection. And then the vaccines come in, right? And now the whole world is building a lot of immunity really quickly, which leads to, on one hand, it leads to, disease is now a lot less severe because we all have immunity.
Right.
But of course, many of us have immunity, either from vaccination, from infection, from both, right? But this now creates a different selection pressure on the virus, right? Because now the main thing is not whether you can bind to ACE2 or you have your polymerases optimized or whatever for being quote unquote human. It's that there's an immunity wall that you need to break through. And that's what we then see with the next, crop of variants, right? Delta is sort of kind of a hybrid example of that. But certainly Omicron is the first big example of now we have a variant which clearly is, quote unquote, immune escape, right? We're saying like that one could break through these immune barriers that existed out in the population and take off from there. And then we have seen that process continuing, right? And then we see this in flu viruses and many others. But that goes back to like Jesper, as we're talking about something like emergence, right? When we're talking about SARS-CoV-2, we didn't have any immunity to SARS-CoV-2. But imagine that SARS-CoV-3 comes around, right? And is closely related to SARS-CoV-2. Now we might very well have cross-protective immunity because we have those SARS-CoV-2 vaccines, we have infections, we have all the population immunity, which means that the likelihood of a SARS-CoV-3 emerging and causing a pandemic in the human population is now relatively less, certainly as we're talking about more severe disease, because we may already have a good amount of cross-protective immunity, right, because of that existence. And of course, the same is true as we're talking about the animal reservoirs, right? Like SARS-like coronaviruses, for example, are rampant in bats, right? But some of the potential reservoir host, intermediate hosts of these viruses may also have immunity. And that matters as we're talking about what's the risk of emergence, what kind of evolution are we going to see. And it's because of that, virus evolution and immunity just plays together really strongly, right? Because the immunity is such a strong selective force on the virus. And of course, it's not new. That is exactly what we see with flu, and this is why we get the annual flu vaccine. And that has to be updated, right? Because flu drifts and sometimes it shifts and you have a completely new makeup of flu, right? It's for exactly these same reasons.
Yeah. So in addition to these immunologic drivers, though, you talk about anthropogenic drivers.
Right.
So there are other things that influence whether these spillovers happen ever to begin with, right? And so talking about if it does spill over, how the immunity impacts whether it becomes pandemic or whatever. Yeah. What about the spillover part? Like what kinds of things influence that?
Yeah, so all of it. So again, immunity is a really key component here, right? Because it all comes down to like, what is the likelihood of you being able to infect a potential host, right? And of course, if that host already has immunity, if there's neutralizing antibodies, you might get into that host, but you may not be able to infect them because there's enough neutralizing antibodies to take care of the virus before it gets a chance to cause infection in that host, right? And this now we're talking about any host, whether it's a human host or an animal host, right? And of course, as we're then talking about like, okay, but now imagine that you have a virus, it has managed to get into the human population, But there is a lot of immunity in that population, which means that the reproductive number of the virus never manages to get above 1, which is when you get and you see a wave and you see exponential growth and you have an outbreak, right? Is that because there might be enough immunity in the population here to keep that reproductive number, and now it's the effective reproduction number because you have immunity, not the basic one, down below 1, such that you might see a handful of cases, but then it might die out from there simply because there's too big immune barriers in the population. And I say population, but that population can be, the human population, or it can be a potential intermediate host animal population from which it might then jump into humans from there, right? The same thing is true there, that they may also have immunity, right? And of course, that is one of those barriers that viruses like receptor binding and all these different things will also have to cross.
And I guess some of the things like where people are living, their proximity to animals, the way we are farming, the way we are deforesting, the way we are traveling around the world also influence how frequently we contact these potential reservoirs, and that probably also has some influence on spillover.
Yeah, so
right?
this is a huge one, right? And this is because, like, when we're talking about, there's the spillover itself, right? But then there's the establishment in the population, and then there's an outbreak and potentially a pandemic from there, right? With SARS-CoV-2, all of those things happened.
Yep.
Right. And the problem is that the way in which, like, things like having, live animal markets, for example, where, you're selling animals that are chock full of all sorts of different viruses, right? That many of which they catch on the way into those animal markets, which is an absolutely terrible business of, animals being stressed and put together, all sorts of different animals from all sorts of different places, right, and get into transportation channels that ultimately end up in wet markets, for example, right, in large urban dense centers, is that all the animals are going to have viruses, right, and some of them are going to have potential human ones or human emergent viruses, right, and we know this. And of course, as population expansion and encroachment into forested areas in which animals currently live, and it's just the animals, of course we're going to create those connections more and more often. Yeah. Right. And that just means that we get into contact with these animals and therefore their viruses that they carry more often. And that's the spillover part of this. Yeah. But the additional part is like, okay, but you have a spillover. But for example, if a spillover with something like SARS-CoV-2 happens out in the rural areas, chances are that it might cause maybe a handful of infections, right? But then will die out. Now, we think of SARS-CoV-2 as a very transmissible virus, and it is, right? But it's very clustered. And in fact, in places in which you have a very low human density, the likelihood of this causing and the next infection is much less. Yeah. Right. And typically this will just result in these dying out.
They just burn out.
And so they just burn out, right? You're saying it might cause one infection, it might cause a handful of infection, but then no more infections. And this probably happens frequently as we're talking about SARS-like coronaviruses, flu viruses, and other viruses out in the rural areas in which they may get into contact with these animals. But they're too, low density for the virus to take off in a human outbreak, for example. And that comes into the second part, which is like, yes, but if all of that happens out there, and then we take those animals that we get into contact with, and then we take
bring them into the
dense
urban centers, seems like a bad idea.
Is not a great idea, right? Because now you have the density required that if you do get those viruses coming into these areas, now they're much more likely to take off in an outbreak, right? Early outbreak. And then that brings the last part, which is the travel part, right? Which is things like, and then we have global travel, right? We all, traveling around the world, right? All the time, which means that something like SARS-CoV-2 can travel around the world extremely quickly, right? So something you might see in China, or something you might see in somewhere in Southern California, right, or a new flu pandemic or whatever that might be, right, is that getting from there to all over the world is in a matter of
hours,
just a few days,
right?
Yeah. And of course that's new, right? And as that we get more and more people, and as we travel more and more, of course the risk of this it's much higher, right? And it's funny when you sort of like think about it that way, because and I get this question often when I give a talk, where sort of people think of pandemics as something pretty rare, and maybe it happens, maybe it doesn't happen. And I give a talk and I talk about this, and people are like, how is this not happening all the time?
It probably is, and we just don't know about it, right?
So again, the spillovers and all these viruses are trying to come in all the time, right? It's just that there are so many steps that all needs to line up, right, before it then can take off in a pandemic, right? But of course, the ability to do that as we encroach in animal areas, as we have human expansion, as we all these different things, only increases, right? Yeah. Which is why the risk of severe pandemics like we have seen with SARS-CoV-2, for example, is ever increasing. And of course, one big thing that we have not mentioned yet, but things like climate change, right, where for example, the vectors and the reservoirs carrying these viruses, those also expand in range. And that means that we're going to see, new viruses in new areas. And we're already seeing this with, local malaria cases in Florida, for example, and, West Nile all over Europe and like other places in which they haven't had these viruses for quite a while, and now they're re-emerging or emerging, right, because of the expansion of the vectors themselves.
So studying all this, what can we learn? This seems like inevitable.
Yeah.
Everybody says it's inevitable. There's going to be another pandemic. We don't know if it's next year, 10 years from now, whatever. Right. What have you been working on, and what do your studies show looking at this virus evolution? What can we learn from that to try and prepare if we can get those who are responsible for preparing to do what they need to do?
I think the main thing, right, is it's sort of a multifaceted approach, right? And I'm looking at spread and evolution and emergence, and I work with colleagues who work directly in public health, for example. But the fact is that no one of us can sort of okay, here's what we're going to do, and then we'll lower the risk of pandemics, right? It requires a lot, right? It requires, for example, as we're talking about the risk of having, wet markets, for example, and having things like animals brought into dense urban areas, and that poses a risk, right? Which is true, it does, right? But then the question is okay, so knowing that, what are you going to do about it, right? And I think many people maybe will say, let's ban it, right? We can't ban the trade. It's crazy that we even do that. And I agree with that, right? But the problem is that you by banning it, you might not effectively stop the trade. You're just going to move it on the ground.
And then you can't.
Right? So now you can't actually monitor anything because it's still happening. It's just now it's much harder to detect, right? So the question is saying, so maybe that's not the best approach, right? But you could say that it's okay, so given that, and given that a lot of people's livelihoods are, dependent on the trade, these kinds of trades, right? And so is the food source too, right? Is but maybe we get a better regulation around it, for example. Mm-hmm. Maybe we can put steps in which we're saying look, we're going to test for these different viruses before they come into the markets. Maybe we're going to ensure that they have better husbandry. Maybe we're trying to saying let's not mix those animals with those animals, things like that, right? Which are simpler to do, although not simple, right? But could be done. So we can do that, right? And then, of course, we can invest in things like surveillance, where we're saying, okay, if a new pathogen emerges, we want to know about it as soon as possible. If all of a sudden we see an outbreak over here, we need to make sure that they're going to report on it early, and then tell it to entities like the WHO, right? And the whole world knows about it, which means that we need to incentivize early reporting, for example. Because again, because of the travel, as we talked about, right, that an outbreak that happens over here and has pandemic potential, it's not going to be an outbreak, or it's going to be all over the world very quickly, right? And there's nothing we do about that. So there's all these different things that we need to do. And then, of course, continue to invest in science around vaccines and diagnostic tools and drugs and all the different things that we know need to be done, such that, when the next outbreak happens, maybe we can limit it at the scale of an outbreak before it becomes a pandemic. Or if it becomes a pandemic, that we have, the vaccines and we have the drugs, then we can line them up pretty quickly. The problem, of course, is that we're not doing any of that, right? In fact, we're doing the opposite, right? And that to me is I worked on pandemic preparedness and sort of responding to outbreaks and epidemics in multiple different parts of the world.
Right.
And I don't think I quite expected that, right? I mean, I thought maybe it's like, okay, in a future pandemic, we are going to do a lot of right things and we're going to make a lot of mistakes. Then we learn from those mistakes, right, and fix them. And then the next time we're going to go do better, right?
That's the scientific way.
Maybe that's the scientific. I mean, now I see it as a very naive way, right? And I even remember, like early on in the pandemic, I said this to my wife where we were talking about what was happening, and I said, "Look, I think the first wave, I think, is going to be really bad, because I don't think that we actually understand what we're dealing with here, and we're not going to react in the way in which we need to react, because we were talking at the time, like, entities didn't want to test for it. There was like all these different things. It was like, so as I told her, it's like, I think it's going to get really, really bad. But the good news is that I think, like, so that's going to happen, but then we're going to learn from the things that we did wrong during the first wave. And then we're going to improve on it in the second wave. And then I said, maybe the second wave will also be pretty bad, but then I'm pretty sure that we'll sort of get a handle on it. Right now, I was right about the first part. Yep. Right, it was pretty bad. Right, I was very wrong about the whole like learning from the mistakes. And I simply think it's because, I mean, in hindsight, of course, we were not going to learn from these mistakes. But so it's probably naive, right? But it is crazy that we come out of a pandemic like this, and then you're saying like, yes, and because of that, let's now stop all our research on vaccines. Let's not take vaccines anymore from vaccine-preventable diseases. Let's, destroy much of the research that's focused on understanding these viruses and where they're coming from and what to do about it. Let's take our public health agencies and have layoffs and ensure that the people that actually, try and monitor and respond to infectious diseases, we're not going to have them anymore because we don't want them anymore. That to me is insane. Right, and that I certainly had not foreseen, like as a result of the pandemic, right? But that is exactly what we are seeing. Yeah, and that's a problem, right? And it also leads us to this situation that we find ourselves in, where, immediately following a major pandemic, you would think that your pandemic preparedness apparatus would sort of improve as a result of that. And I think technologically it has. Right, there's a lot of new tools and a lot of new things like mRNA vaccines, for example, that were developed during the pandemic. It was a huge success, right? Is that we have all of those technological tools, but of course it matters little if people don't want to take them, or if we can't get them out to people and we can't get them to the world, right? And that's exactly where we find ourselves today, I think.
It's incredibly frustrating.
It's very frustrating. Yes. I mean, if I had the solution to that, then, I'll go do that. I honestly just don't.
Yeah, I think we all feel really frustrated and sometimes hopeless, but I think there's still hope.
Yeah, I don't think it's hope. I don't think it's hopeless. I think, I think it needs to start with the recognition of what's actually going on. Yeah. Right. I mean, I do think it's important that people understand that, what is happening in terms of, vaccine recommendations and everything that's happening here is an anti-vaccine agenda, right? And it is to a large extent an anti-science agenda. And we need to understand that is what is happening. Yeah. And we can't sort of just sit back and hope that magically it will just resolve itself, right? Similar to the early stages of the pandemic, where we can't just sit and then saying like, oh, but it's not going to come here, or the virus was just sort of go away, right? It's up to us to ensure that change happens. Yeah. And whether that's responding to a pandemic with developing vaccine fast and diagnostics and all these other things, right? Or whether it's actually trying to work against some of these forces of anti-vaccines, misinformation, disinformation, anti-science, is up to us to do that.
And when you say us, you mean the scientists.
The scientists, the scientific community. Yeah. Right? The public health community, the universities, the academic communities, right? I mean, we can't be out here fighting the fight, alone, right? We really do need to do this together, right? And I think the first aspect of that requires recognizing what's going on and talking about it in the terms of what is actually going on, which again, anti-vaccines and anti-science, disinformation, misinformation, right? And then what each of us individually do, of course, that varies based on individuals, right? Some people might be able to do a lot. Some people might have a voice. Some people might be very good at communicating around this. might help educate the public and politicians and others, right? And others might be more sort of like, on the backseat of it, saying like, I don't quite have the energy to it. I don't see myself. Well, that's fine too, right? I mean, we all decide to do this. But I think, again, like trying to do it alone, with individual universities trying to fight, individual scientists trying to fight, that's never going to work.
I agree.
Right?
I think one of the more important things is to get the scientific community together.
Yes.
And I think part of that needs to be recognizing that those who can communicate and do communicate, that should count for something.
Oh, it certainly
right.
Count for something.
We should not be eating our own, right? I feel like a lot of scientists, some administration, not here where I am. I feel very privileged in that way. Yeah. But a lot of my colleagues are like, no, I'm not going to speak up. I get no credit for it. I'm not bringing in a grant. I'm not doing this. But if we don't, who will? Yeah. Right. So if we don't try and contribute to the narrative, who is controlling the narrative?
Who's supposed to do it? Right.
Right.
If we can't talk about it, who's supposed to do it? Somebody else?
Yeah.
Right. I mean, that's the part that, and again, I'm not saying that everybody should, because again, there are scientists that don't feel comfortable talking about these things and I totally get that, right? Because many scientists just want to focus on the research, and that is totally fine, right? Like, I'm not holding that against anybody for not speaking up, for example, right? Because I get it.
Yeah.
Right. And I think that's important too, right? But I also think when it comes with like the engagement with the public, for example, a lot has been said about, the mistakes that scientists made during the pandemic, and, we needed to explain what's happening better and all these different things, which I completely agree with that, right? Like, to the point of like, scientists need to communicate better. It's like, yes, we can always learn how to communicate better. But at the end of the day, that's not the major problem here, right? The anti-science, anti-vax agenda, that's the problem. And will it help if scientists learn to express themselves better, explain the science better, the caveats better? Sure, of course, every little helps, right? But we also need to tackle some of these bigger problems. And of course, the scientists that do then decide to stand up and speak up need to be supported in that mission by the institutions, by the colleagues, right? And need to be rewarded for doing so, right? Because frankly, it is an important part of doing science these days, right? Is that we can't do this in a vacuum, and we can't think that this happens in a vacuum either. Of course, the public is going to care about it and should care about it.
They should. They're funding it.
Yeah, they're funding it.
Well, we hope they will continue to fund it.
No, I mean, science is such a privilege, right? Because it is one of those things where you can pursue your passions, right? And you can do important work, and it is publicly funded to a large extent. Right. And it also just happens to be financially, economically, a really good trick, right? Because it brings a lot of back into the society, right? In terms of, and you look at the United States, I mean, science and technology is what we do really well, right? And we should really support the continuation of this because, 10 years from now, don't we still want to be the world leader in this? I mean, I would like to, right? I think that would be beneficial, right? And again, like with a lot of what's happening now, I'm certainly worried that is not necessarily what we're going towards right now.
Yeah. Switching gears a little bit, coming back, so we got a little bit off there. Coming back to the viral dynamics a little bit. I'm not looking so much at a population, but at an individual.
Right.
Some of the other things you talked about in the article were about how an individual's immunity and their antibodies also help shape viral evolution. Can you say a little bit about that? Yeah.
So this is where, as we are thinking about just viral evolution in general, right? We need to think about like, what are we thinking about? Are we thinking about, for example, a person living with HIV, right, who has an HIV-1 infection? I was thinking about how that virus might evolve over time in this individual, and maybe that determines some of the drug treatments the patient is receiving, right? Because over time, you may start developing resistance to one drug, but of course you have combination drugs, right? So there, following viral evolution over time in the individual patient can be very important for clinical care. When we're talking about something like, all viruses evolve in individuals, right? And that's
I don't think that's intuitively obvious for everyone.
No, maybe not.
So like you get infected with one virus, it's actually going to mutate within you,
correct,
and fight against your immune system.
Correct. Yes. So this is the the virus evolution happens inside individual as it's replicating, right? And the virus doesn't really care about you, right? It just cares about itself, right?
It's just trying to get out.
It's like, well, I want to keep replicating here, right? And of course, it also needs to transmit to the next host, not because the virus knows it needs to do this, right? But if it doesn't, it all stops there, right? But what happens inside the individual host obviously depends on how, what is your immunity at the time of the virus infecting you? How long is the infection, for example? If you're a naive host, right, with no immunity, and the virus infects you for, 3 days or 4 days, right? In terms of, adaptive immunity, for example, is not going to pose a selection pressure on the virus, right? Because there simply isn't enough time for that B cell response to develop, or the T cell response if it's humoral immunity or cell-based immunity, right? That's different in these chronic infections like HIV-1, for example, is the one I mentioned, right? Because there is a lot of T cells that recognize HIV, there's a lot of it, exactly, there's this continuous pressure, right? What is interesting as we're talking about, SARS-CoV-2 and COVID, right, is that, of course, now we all have immunity, whether vaccine-derived, whether infection-derived, or a combination of both, for most people probably these days, right? Is that poses a selection pressure on the virus as it infects you. But importantly, too, is that as it tries to transmit to the next host, right, is that some virus that is good at, evading immunity within you may also be good at evading immunity in the next host, and may ultimately have a fitness advantage at the population level, right? But it also needs to be able to transmit, right? So if it's saying like, well, for example, you have chronic infections with SARS-CoV-2, which is where we think these variants of SARS-CoV-2 is coming from, probably chronic infections, right, that might last for months, probably in somewhat immunocompromised individuals to a large extent. And during this period of time, the virus is evolving within that host, right? And immune selection and immune pressures is going to be a huge part of that, right? Because the host is continuously trying to target the virus, and the virus is continuing trying to evade that, such that it can continue that chronic infection. But of course, there is no transmission there, right? So during this period of being, maybe it's very good at evading immunity within this host, it may have forgotten how to transmit to the next host. And transmission is, stability of the virus, for example, is a key one here, right? Is that it might simply just lose that just because it's now no longer being selected for just living in a
just because it has an opportunity to make more copies of itself,
correct,
which is its only goal.
That's
so if it's
goal.
Doing that in that individual, there isn't really a pressure to transmit to a new host.
There's not that selection pressure for the transmission, because during this process, it's not doing that, right? But of course, if it loses that ability, right, then you're not going to see it, right, because nobody else is going to get infected. Now, when we look at something like Omicron, is that Omicron probably initially might have been sort of de-adapted for the transmission aspect of it, but it was really good at immune evasion, right? And what was also true at the time was that we, basically all of us had been, either vaccinated with the same vaccine antigen, the original version of the Wuhan strain of SARS-CoV-2, or we have gotten infected by a very close relative of that, a variant of that was very close to that, right? So our immunity was very similar, whether it was, your vaccine or my vaccine or somebody's infection versus my vaccine, right? Is that for that was very, very similar in the population, which means that if you're then that virus that evolves, around my immunity, for example, as part of a chronic infection in me, chances are that you would be good at also evading the immunity in somebody else, because there's a lot of sharing of the epitopes and everything that's being shared in terms of what's targeted, right? And that's probably why Omicron was so explosive, because Omicron was really good at that, right?
Yep.
Which means that it got to the next individual, It's like, oh yeah, this is the bit that I have really learned how to go around, right? So I'll just keep going. And Omicron just exploded, right? And in fact was by far the biggest wave, right? With the most death following that too. So that obviously happened. But now if you're looking at what's happening today, all of this is now different, right? Because now the sort of immune history that we have in the population is much more diverse, right?
Okay.
Because maybe you got that booster, maybe you got that booster, maybe you got infected in April, maybe you got infected in November, maybe you got and over time, this just means that if you're looking at your SARS-CoV-2 immunity versus my SARS-CoV-2 immunity, in terms of the antibodies that we have and the T cells we have, right, they probably recognize quite different and distinct epitopes, right? Which was less true in the really early stages of the pandemic. And that is probably one reason why we haven't really seen the Omicron event, right? And it's probably also the reason why somebody like me can be a little bit confident that maybe we are not actually going to see something that explosive, just because there isn't a really easy evolutionary path in which a virus could come in, SARS-CoV-2 come in at a new variant, and saying yep, I can evade all of this immunity.
Find a new path to take, yeah,
because
most of those paths are going to be blocked to some extent. Yeah. Oh, that's really fascinating.
And I think this also speaks to, I've been talking about this concept of broadening immunity for quite a while, right? Where, I think there was because we need annual vaccines. I think we need an annual flu vaccine. I think we need an annual COVID-19 vaccine, right? I do think they differ a little bit when we need them. I think we need the COVID-19 vaccine over the summer. I think the flu vaccine later, in the fall, early winter, right? Just to sort of time what we're seeing in terms of waves. But we need that, right? But that's not to say that we need to have the vaccine for COVID-19 needs to exactly match the variant that is the most prevalent at the time of getting the vaccination, because we simply just can't do that. It outruns us every time, right? It takes too long. By the time we have that vaccine ready, the virus will have gone on to the next evolutionary step. But over time, of course, is that as you're getting a multitude of different vaccine antigens with annual vaccines, is that the hope is that you'll broaden your immunity over time. And populations, importantly, will broaden their immunity over time, both via vaccination as well as, of course, the infections that also happen.
Presumably, if you are vaccinated, you're still going to get exposed.
You're still going to get exposed.
Tickle your immune system, and you can start to broaden that within yourself as well, from natural
and
exposures.
You will because again, as we know by now, while the really early stages, the vaccine for a short period of time really did protect very well against infection.
Yeah, it did.
Right.
And I think a lot of people deny that. They ignore that fact. But there are data out there to show that it was actually preventing infection for a little while.
And I would say, even today, I also think, because in a lot of this, people sort of look at it, in black and white, either they work or they don't work, right? But that's not how it works, right? It's not it's either 100% effective or it's 0% effective, right? It's in between those things. And so to the question of, will vaccination lower your risk of infection with SARS-CoV-2? Even today, the answer is yes, it will, right? If you take 8 individuals that got the recent boosters, or 10 individuals of the recent boosters, and you expose them all to SARS-CoV-2, right? Maybe 3 of them will get infected. You do the same in a population where 10 individuals have not received the booster, right? Maybe 5 of them will get infected, right? In both groups, people will get infected, right? But they're less in one group in the other group, which means that you lowered your risk of getting infected in the first place. The other thing you lower is that but maybe you do get infected, but now your immune system is ready to respond pretty quickly, right? Which means that it'll probably resolve that infection faster than if you hadn't got it. And of course, ultimately, it means that the kind of disease that you're going to experience as part of that infection is overall going to be less. It doesn't mean that you can't get severe disease, right? It doesn't mean that you can't get COVID and feel really awful. And maybe you would think I got my vaccine, I got COVID, and I still felt like crap, so the vaccine didn't work. It was no, the vaccine still worked. And yes, you did feel like crap, but chances are you felt a lot less crap than if you hadn't gotten your vaccine in the first place. Right. And again, this comes back to it's not black and white. It's not either 100% or 0%. Right. And that's why I think moving forward, at least as it is now, right, is that we will continue to get exposed to COVID. We will continue to get infected with COVID, even if we continue to keep up with our vaccines. Right. But keeping up with the vaccines just means that we can continue to broaden that immunity. We can continue to lower the burden of disease, even if infection levels stay somewhat high. And that is also important, right? Because people that can't get the vaccines, that don't have good immunity, right? Immunocompromised or whatever, by us continuing to do this, we will also lower the number of just infections out in society in general, right? And that's going to help them lower their own risk.
And one other piece that I didn't hear you say necessarily, but when you have that immunity on board and you get infected, not only lower your disease, but you also lower the number of replications of the virus in that individual. So they have less of ability to transmit. So it's still that idea of if it reduces transmission, even if it doesn't reduce transmission on paper necessarily, the fewer viral particles that you produce and breathe out, the less likely you are to infect your neighbor.
I mean, again, it doesn't lower it 100%.
Right.
Right. So you can or you cannot transmit. Right. It's just it goes down. Right?
Every little bit counts. I mean, everything we can do to reduce it.
That's right.
Helps.
You know, when you have a beer, it's not either full or it's empty. Right.
Well, it can be.
It's like, well, it's somewhere in between, right? It is full in the beginning and it's empty in the end, right? But in between, still you still have a beer. Right?
Yeah.
And I think like the same is true as we're talking about immunity here, right? And especially as we're talking about, you know, for example, this scenario in which maybe you just got your annual booster, right? For example, last year I got my annual COVID-19 vaccine. And I got COVID for the first time, I think 3 or 4 weeks later, right? And I was like, oh man, you know, seriously, right? And I was really bummed, right? Because, you know, I was pretty sick, right? I wasn't feeling great, and I had it for like a good 15 days. I was like, this really sucks, right? So, you know, I wasn't happy about it. And, you know, I was supposed to travel to Africa, and we had the opening of my, you know, colleague Kristian Happy Center, and I'd been looking forward to this literally for a decade, to see him open his center, right? And I couldn't go because I had COVID and I felt terrible. So, you know, you can see like, this is the example is like, man, I just got that vaccine and then I got COVID anyway. And it's like, yes, but on the other hand, if I hadn't actually gotten that vaccine, I would probably have been much, much sicker than I ended up being.
Yeah.
Right. And it still sucked, right? But it wasn't that, you know, I wasn't dying. Right. And I would rather it being like nothing at all. Right. And it was still severe enough, right? Yeah. But that's really important to remember. And of course, like in this case, like, you know, you got your vaccines, you have the antibodies are ready to go, right? So it's not that you have to spin up your memory T cells and wait for them to turn, you know, to produce new antibodies. You already have a lot of those that will help take care of the virus immediately, right? It won't be enough. Like, again, in my scenario, I still ended up being infected and being sick, right? Yeah. And of course, the same is true on the T cell side, right? So the more ready that is, the more cells you have, the more ready they are to go, the better it is. Right. Again, it does not mean that you lower the disease to zero so you don't see it at all. Right. Yeah. It's less. Right.
I could talk to you for a long time. This is like a super fun discussion. I always try to like one like normal human type question. Right. I have lots of different ones that I ask, but I have a fun different one this time. Okay. So many people don't know a scientist. Right. And they actually might think scientists just always hold up in their labs and they have no hobbies and they don't do anything. But we all put our socks and shoes on in the same manner. So what makes you an everyday person? I
mean, I think like the everyday person of me is that, you know, I haven't always been a scientist, first of all. I mean, I used to have like what I think of as normal jobs, right? I mean, I used to be working at the post office and, you know, I had a paper route and had all these different things. So from that perspective is that I think it makes me normal, but I also have normal hobbies. I mean, you know, I like hiking a lot, for example. I like climbing, you know, with my wife and with friends, right? And we also like hanging out in the dirt. I mean, we go, you know, we live in California and, you know, we take the Jeep into the deserts and into the mountains and we camp out in the dirt, right? Like any other person. And we enjoy that a lot. Yeah. And I don't know how many scientists do that, but, you know, I like it for the nature and the peace and quiet. So, you know, I think that's pretty normal. And then we go back to the lab on a Monday morning, right? But I think that's important, you know, for me, myself, and I like to think that most people in the lab too do have hobbies. Obviously, it's San Diego; a lot of them are surfing, right? But there are other things. There are other things too, and I certainly encourage that because I think it's important. Right? Yeah, it's important to have other things too. Oh yeah, for sure. Yeah, like, you know, and it just, you know, it also just gives you this clarity of mind, right? And sometimes, you know, I have sometimes in the past, you know, go on a hike and all of a sudden I start thinking about scientific questions maybe, and it sort of gives me like, the fresh air, and I was like, oh yeah, maybe I'll think about that a little bit more. And
fuzzy thinking. When you're not thinking about it, you're thinking about it, and creative ideas.
You lose the creativity.
Yeah.
Right?
Cool. Yeah. Well, thank you again for coming here. This was a fun conversation. Great. So that's a special episode of Immune, an immune booster. If you like these shows, please consider supporting the shows of Microbe TV. Immune and all these other shows are able to go on because of your contributions. Microbe TV is a 501c3, so all of your contributions are tax deductible. So if you want to do this, just go to microbe.tv/contribute. I'm Cindy Leifer at Cornell University. You can find me on Cindy Leifer on Blue Sky. And thank you again to Kristian Andersen for coming and talking to me. Great. Music on Immune is by Tatami. And I want to thank you for listening to Immune, the podcast that's infectious.